What is micropropagation? Micropropagation is the artificial process of producing plants vegetatively through tissue culture or cell culture techniques. In this artificial process of propagations, plants are produced in vitro by asexual means of reproduction or by vegetative propagation. Micropropagation is also known as, we call it, tissue culture. So, plant tissue culture is a collection of techniques used to maintain or grow plant cell, tissues, or organ under sterile conditions on a nutrient culture medium of known compositions. Besides, tissue culture is a type of asexual reproduction. What is application of micropropagation? Micropropagation is method produce plants free of disease. Hence, disease-free varieties are obtained through these techniques by using meristem tips culture. So, proliferation of in vitro stocks can be done at any time of the year. Other than that, a nursery can produce fruits, ornamental, and tree species throughout the years. What is the basic concept behind the application of micropropagation in technology? First is totipotency. Totipotency is the property of live plant cells. They have the genetic potential when cultured in a nutrient medium to give rise to a complete individual plants. Second, differentiation. Differentiation is the process of biochemical and structural change by which cells come specialized in, from, and functions. Next is redifferentiation. Redifferentiation is the further differentiation of already differentiated cells into another types of cells. For example, when the component cells of colors have the ability to form a whole plants in a nutrient medium, the phenomenon is called redifferentiation. And lastly, the differentiation. The differentiation is the phenomenon of the revisions of matter cells to the meristem state, leading to the information of colors is called the differentiation. So, in these two phenomena of the redifferentiation and the differentiation are the inherent capacities of living plant cells or tissue, and it is described as totipotency. So this is some of example of plants that are developed through micropropagation on our plants such as banana, tomato, pine, eggplant, pineapple, yam, sweet potato, and lastly is carrot.
Next, there are a few groups that utilize such plant biotechnology. First of all is banana. Banana are typically propagated vegetatively, and micropropagation plays a crucial role in their cultivation. The conventional propagation method, which is involved using sucker and offset from existing plants, poses the risk of carrying diseases or pathogens. Micropropagation offers a solution by producing disease-free banana plants in a controlled environment through tissue culture techniques. The process involves taking a small piece of the plant, often from the shoot tips or meristem, and culturing it in a nutrient-rich medium under sterile conditions. Hormones are used to encourage the development of shoots and roots. This method allows for rapid multiplication of banana plants. ensuring they are disease-free and genetically identical to the parent plant. Next, potato also utilize suspend biotechnology. Potato cultivation heavily relies on seed tubers for propagation. However, traditional seed tubers can carry diseases such as virus and bacteria, leading to reduced yield and quality. Micropropagation via tissue culture techniques helps in producing disease-free potato seed tubers. The process involves using small tissue segments or meristem from healthy plants to initiate cultures in a sterile medium. These cultures are then multiplied in a controlled environment, generating disease-free planters. These planters eventually develop into whole plant and are used as disease-free propagules for cultivation. The other one is carrot. Carrots are primarily grown through seeds, but micropropagation techniques in carrot cultivation are less common compared to the other crops. Carrots plants are biennials, taking 2 years to complete their life cycle, and they are typically grown from seeds. However, micropropagation has been explored for specific purpose in carrot production. Although it is not as intensively practiced as some other crops, firstly, it helps in disease-free plant production. Micropropagation can be used to produce disease-free carrot plants. By using tissue culture, small sections of the carrot plant, like the shoot tip or meristem, are cultured in a sterile, nutrient-rich medium. This process helps in generating disease-free plantlets that can be grown into mature plants. Besides, it is rapid multiplication. While carrots are predominantly grown from seeds due to their relatively simple, inexpensive propagation through traditional means, micropropagation can offer a way to rapidly multiply specific carrot varieties. This technique can help in quickly producing a larger quantity of plants with particular traits or characteristics. Moreover, micropropagation in carrots might not be as widespread as in certain other crops due to ease and cost-effectiveness of traditional seed propagation. However, it still holds promise for specific application, particularly in producing disease-free plants, preserving unique genetic material, and advancing research in carrot genetic and traits. As technology and techniques in plant biotechnology continue to advance, the application of micropropagation in carrot cultivation may expand further. Next is sugarcane. Sugarcane is a vital crop in the production of sugar and other related products, and micropropagation techniques in plant biotechnology play a significant role in its cultivation and improvement. Although sugarcane is traditionally propagated through stems cutting or set, micropropagation offers several advantages in the production of disease-free planting material and the improvement of sugarcane varieties. Firstly, it is disease-free plant production. Sugarcane is susceptible to various diseases including virus, bacteria, and fungal infection. Micropropagation helps in production of disease-free sugarcane plant by utilizing tissue culture techniques. Meristem or shoot tips from healthy and disease-free donors plant and cultured in a sterile nature medium. This method ensures that the resulting plantlets are free from pathogens, providing clean and healthy planting material. Besides, it is rapid multiplication. Micropropagation enables the rapid multiplication of sugarcane plant with desirable traits. Through tissue culture, a small piece of sugarcane tissues is used to initiate cultures in a controlled environment. Growth regulators stimulate the development of shoots and roots, leading to the generation of multiple plantlets. This method accelerates the multiplication of selected high-yielding or disease-resistant sugarcane varieties. Grapes are another crop that benefits from micropropagation techniques. Elite grape varieties with specific characteristics can be rapidly multiplied through tissue culture. This method allows for the production of disease-free, genetically identical grapevines on a large scale. In great micropropagation, shoot tips or meristem are excised from selected vines and cultured in a nutrient medium. Hormones are used to stimulate shoots and root development, leading to the generation of healthy plantlets. These plantlets can then be acclimatized and transferred to the field for further growth and grade production. Micropropagation in these crops contribute significantly to disease management, genetic preservation, and the production of healthily, uniform plant, ultimately enhancing crop yield and quality. The last group that utilize such plant biotechnology is tomatoes. Tomatoes are intensively cultivated worldwide and are among the key crops benefiting from micropropagation techniques in plant biotechnology. Micropropagation plays a crucial role in tomato cultivation due to several reasons. Firstly, it disease-free plant production. Tomato plants are susceptible to various diseases and pathogens, impacting their yield and quality. Micropropagation helps in producing disease-free tomato plants by starting healthy tissues or meristem and growing them in a controlled, sterile environment. This technique ensures that resulting plants are free from infection, virus, or other diseases commonly transmitted through seeds or vegetative propagation. Next, hybrid seed production. Micropropagation contributes to hybrid seed production in tomatoes. It allows for the maintenance and propagation of parental lines used in hybrid tomato breedings. Micropropagation ensures the purity and genetic integrity of these lines, enabling the consistent production of hybrid seeds with desired characteristics such as disease resistance, improved yield, or better fruit quality.
Now, let's talk about types of micropropagation. There are 5 types, which the first one is meristem culture. As we can see, we use the shoot tip and soak into a distilled water. Then, we took the apical tip into the medium and incubate for 16 hours at 25 degrees Celsius under fluorescent light. Then, transfer into the pots. In this method, a disease-free plant can be produced and can be successfully used for rapid multiplication of various herbaceous plants. The second one, we have callus culture. In this method, for example, we use a carrot. The plant tissue will be placed in an artificial growing medium culture until the callus is formed, like the one shown in the diagram. After callus is formed, they are transferred into a culture medium containing plant growth regulators for its induction of adventitious organs. After a few weeks, a new plantlet is exposed to the environmental conditions. The third is suspension culture, which in this method we sterilize the material and the material are dispersed to allow them to grow in an aerated and sterile liquid culture medium. Lastly, disintegration will occur. The fourth one, which is the next one, is the embryo culture, which is the next method. In this method, we will choose the immature seeds to be sterilized. Then, we will peel off the glands, and the glands were placed into a culture medium with proper nutrient in aseptic conditions or in a free-form contamination by pathogens. The embryo will grow into a seedling until the plantlet is ready and they could be transferred into a pot. Last but not least, the last method is protoplast culture, which the leaf will undergo sterilization and the epidermis will be peeled. The peeled leaf will be mixed in the enzyme mixture and the protoplast will be removed. Wash the protoplast, then the culture will turn into callus. The cells will develop a cell wall and increase the self-division and grows into a new plant. So, this is the stages of micropropagation from stage 0 until stage 4. The initial stage of micropropagation is the stock plant are selected and grow under controlled conditions to make sure it is healthy. The first stage needs us to isolate the explant, sterilize, wash, and establish on appropriate culture medium. By means, they make sure the cell is free from any bacteria. The second stage is shoot multiplication, which involves the rapid multiplication of shoots or rapid somatic embryo formation in a defined culture medium. The third step is rooting of the shoots. The shoots are transferred either directly into the soil for root development or into the nutrient medium in the laboratory. The last one is transferring plantlet in the greenhouse environment. Here, the plantlets are established in the soil and the shoots from the laboratory are transformed to a greenhouse under controlled conditions of temperature, humidity, and light.
Now, I will be talk about the strength and weakness of micropropagation. First of all, for your information, micropropagation has been used in so many countries. One of the most strength in micropropagation is it can be used to produce disease-free plants. The meristematic culture is generally employed in cases where to aim to produce disease-free plants. It has been demonstrated that the shoot apex of virus-infected plants are frequently devoid of viral particles or contain very low viral concentration. It can make the plant less virus and disease-free plants. Second, the production of many plants that are close to each other. It is one of the main advantages of micropropagation in its ability to produce a large number of genetically identical plants, which known as clones, is in a relatively short period. Means that we can make more plants, clone plants in short period. This is particularly useful for rapidly multiplying reusable trees. The third one is space efficiency. Micropropagation allows for efficient use of space, as a large number of plants can be produced in a relatively small area compared to conventional methods. Micropropagation often involves the use of scalar cultures media, such as agar or other natural gels. This eliminates the need of large amount of soil and containers associated with traditional method. As a result, the space required for each individual plant is minimized. Fourth strength of micropropagation is it can produce rooted plantlets ready for growth and save time for the grower instead of cut seeds or cutting. Unlike traditional method that may be season dependent, micropropagation enables year-round production of plants. This can be especially useful for crops that have specific growing seasons. Despite the strength of micropropagation, there is also a flaw in micropropagation for this plant. First, the weakness is the genetic instability. Some plants may exhibit genetic instability during the micropropagation process, leading to variation in characteristics among propagated plants. This can reduce the reliability of the method for certain species. The second one is cost factor. Cost factor of the micropropagation establish the facility that involve high initial costs, including equipment, infrastructure, and skilled personnel for maintaining aseptic condition. This may limit the accessibility of the technique to small-scale growth. Third one is the risk of contamination. During the course of micropopulation, several slow-growing microorganisms such as Aspina species, Bacillus species, is the species that contaminate and grow in culture. The microbiome infection can be controlled by the addition of antibiotics and fungicides. However, this will adversely influence propagation of the plants. Last one is production of disease-free plant. Even micropropagation lead to the disease-free plant. Many plant species are infected with pathogen. Viruses, bacteria, fungi, mycoplasma, and nematode that cause systemic disease. Although these diseases do not always result in the death of plants, they reduce the quality and yield of plants. The plants infected with bacteria and fungi frequently respond to chemical treatment by bactericides and fungicides. It doesn't always make disease-free plants. It also have a flaw in micropropagation. In summary, micropropagation is a key component of plant biotechnology. It has significantly influenced important crops like bananas, apples, and potato. It is enhancing their production and characteristics. This technology involves growing plant cells or tissue. Despite all its weaknesses, micropropagation remains a crucial tool for maximizing its benefits in modern agriculture and horticulture, contributing to sustainable and efficient plan of propagation.